So it's 3:30. So I would like to thank you all who came to Seattle to be part of the meeting in person. It was really a pleasure to catch up with many of you in the previous events and during the corridor. And I also would like to thank the participants who are joining in online. I don't see them, but I believe Priyanka does. So I would like to, can you move this to the next slide?
Yes.
So I would like to welcome 3 new members to our leadership team. So Roberta Albani is our new patient advocate, and she stepped into the role that Elda Riley has vacated. Elda spent the past few years with us and did a fantastic job as a patient advocate, but due to a number of other conflicting, sort of competing expectations, she decided to resign. And we are really happy to have Roberta in this new role. Josh Mammon, many of you know Josh, and he has been involved with many of the SWOG studies. So now he is our official surgery liaison, and I look forward to work with him. We also have a new position, a diversity, equity, and inclusion role that Gayatri Nagaraj from Loma Linda is the inaugural liaison in that role. So welcome, Gayatri. The next item is a real pleasure and an honor to introduce the recipient of this year's Robert B. Livingston Translational Breast and Lung Cancer Lectureship. Bob Livingston was a superb clinician, a clinical trialist who greatly impacted the field of cancer research. Within SWOG, he was the chair of both the Lung Cancer Committee from 1974 to 1997 and the Breast Cancer Committee from 2000 to 2008. And his honor, with the support of his wife Shirley, SWOG established the Dr. Robert B. Livingston Translational Breast and Lung Cancer Lectureship. The lectureship brings a special guest speaker to the SWOG group meeting, and it alternates between a lung and a breast cancer focus. And this year, it was the turn of the breast cancer focused lecture. And this year awardee is no one else than Daniel Hayes. So Dan probably doesn't need an introduction for this group, but it's my pleasure to just highlight some of his accomplishments. He is the Stuart B. Podness Professor of Breast Cancer Research at the University of Michigan Rogers Cancer Center. He received undergraduate, master's, and medical degree from Indiana University, followed by a residency in internal medicine at the University of Texas Health Science Center Parkland Memorial Hospital in Dallas, and a fellowship in medical oncology at Dana-Farber at Harvard. And he has led the breast cancer program at 3 very illustrious institutions, first at Dana-Farber from 1991 to 1996, then Georgetown University's Lombardi Cancer Center from 1996 to 2001, and more recently at the University of Michigan for 15 years from 2001 and 2016. And at all of these positions, he made a real impact and mentored many of you and many of the thought leaders in our field. So, as you know, Dan's research interests are in the field of experimental therapeutics and cancer biomarkers. He really focused and pioneered establishing guidelines for biomarker validation and served as the chair of the SWOG Breast Cancer Translational Medicine Committee. He was also the inaugural chair of the American Society of Clinical Oncology Tumor Marker Guidelines, which are now updated regularly and really changed the field, how we actually integrate biomarkers into clinical research. Dan was also the president of ESCO in 2016. He's fellow of the ESCO, fellow of the American College of Physicians, a past Coleman Scholar, and also was the inaugural recipient of the ESCO Gianni Bonadonna Award in 2007. And I can't resist to tell you that this year awardee is also our very own Kathy Albane, who will receive the Gianni Bonadonna Award at the ESCO meeting this year. So I think this introduction could go on forever, but instead of that, I actually would like to give the podium to Dan, who unfortunately could not be here in person because of a conflicting engagement that he could not cancel at the AACR. So he pre-recorded his lecture, and if technology allows this, then you will be able to see it in a few minutes. So I wonder if I could ask our technical support group to activate the lecture.
I guess honored is the best word I can use. I'm sorry I'm not there in person, but I had previously committed to giving a talk at the AACR, and unfortunately AACR and SWOG are overlapping this year. So I miss seeing all of you and hope we can all be together at the ASCO meeting and even better at next fall's SWOG meeting. I want to say for the third time how really honored and privileged I am to give this talk. Bob Livingston was the paradigm of a great physician, a great scientist, a great leader, a great mentor, and a good friend. And so hopefully I can do him well with this talk. As always, I want to start out and be transparent about my conflicts. Certainly I have conflicts regarding circulating tumor cells. I think all of you have heard me say this. I've received research funding from the companies that have owned the cell search assay through the last 25 years. And my institution has a patent regarding certain aspects of circulating tumor cells from which we receive annual royalties from the current owner of cell search, Menarini Silicon Biosystems. I have received other clinical research from a number of other companies you can see here. In addition, I'm a consultant and on the advisory board of a number of companies, mostly related to diagnostics that are all listed below. I want to start out my talk and just talk about my experience with Bob Livingston. It was a very interesting journey. Prior to 2000 or so, I had met Bob a couple of times, but I really didn't know him very well. We'd been on a couple of ad boards together. But then in 2001, as many of you know, I moved from Georgetown, where I'd been for 5 years, and I'd been at the Dana-Farber for 15 years before that. And I moved to the University of Michigan. That meant leaving CLGB after 15 to 20 years and joining SWOG, a group I knew very little about. At the time, it was led by Chuck Coltman, who had been the SWOG chair for 25 years. He gave me a great introduction. And also, I got to meet Bob Livingston for real, as opposed to just being on boards with him. And Bob had just become the breast cancer committee chair, replacing a number of superb leaders before him. Therefore, he was spreading his expertise in lung cancer and breast cancer across all of SWOG. Bob at the time was working with Peter Ravden, who had been the breast cancer biology committee chair for a number of years, starting back with Kent Osborne. But Peter had decided to step down as the biological committee chair, really just as I was coming in. We overlapped for about a year. And Bob asked me if I'd like to take over, and I very enthusiastically said, yes, I'd be more than happy to, because it allowed me to continue the kinds of things I'd been doing in CLGB before that in regards to correlative and translational science. Over the last 20 years that I've been in SWOG, of course, there have been a number of changes. Chuck Coltman stepped down, was replaced by my colleague here at Michigan Medicine, Larry Baker. He led the group for about 7 years and then stepped down and was replaced by Chuck Beinke, who remains the chair now and has been for almost a decade. Bob selected his former mentee, Julie Graylow, to be the breast committee vice chair, and she did a spectacular job of that, both with Bob and then when he stepped down, and Gabriel Hordovagi, who had never been involved in the cooperative groups, agreed to take over, and Julie continued to be the vice chair. Of course, Julie is now the chief medical officer of ASCO. Gabriel ran things for about a decade and then stepped down and was replaced by Lajos and Priyanka, who remain the chair and vice chair now. And during a lot of this time, I remained the head of what became known as the Translational Medicine Committee for Breast Cancer, stepping down after 16 years of service when I was president of ASCO, and more than ably replaced by Andy Godwin and Alistair Thompson, who've done a great job in building on what we had built and Peter before me, and now taking it into a whole new world. But let's go back and talk about Bob Livingston. On the surface, I think many people on the outside considered Bob a quote chemotherapist, a word I dislike, but Bob spent a lot of his time focused on dose and schedule of different chemotherapeutic agents, taking advantage of what we knew about the pharmacokinetics of these drugs and how they might work, and trying to take advantage of that in translating into better care for our patients. Starting with SWOG 9625, a study that most of you know, where it was a pilot study to look at different dose and schedules of doxorubicin cyclophosphamide with the advent of growth factors. And then that led into SWOG 0012. Were randomly assigned to doxorubicin cyclophosphamide versus weekly doxorubicin and daily oral cyclophosphamide, with the feeling that constant exposure would be a better strategy than bolus exposure. That paper published in JCO. It also led to SWOG SO221, a study led by Tom Budd, specifically looking at dose dense versus dose densest versus dose densest densest. I don't know how else to explain it. Unfortunately, this trial ended up being a negative study. It did not show an advantage to dose and schedule, which surprised me. I really thought it would. Nonetheless, it's been a landmark study. It was really our chief adjuvant study for the 2000s, published in JCO here. And I think it's going to be a terrific setting in which to do a variety of translational medicine studies. In fact, it already has begun to do so. But you know, Bob was really more than a chemotherapist, as we all know. He was a consummate medical oncologist. He embraced endocrinology, and in fact, to the extent that he was the chair when we started SO226, led by Rita Mehta. Rita did a fantastic job comparing anastrozole and fulvestrant as a combination to anastrozole alone, showing not only there was progression-free survival, but importantly for a subset of patients, overall survival, and was able to get that published twice in the New England Journal of Medicine. Most of us would give her eye teeth for a single first author paper in the New England Journal, and she got the same study published twice. Real feather in her cap, and congratulations, Rita. You did a great job running this. And it has now served as an avenue for more translational science. A paper we published recently regarding circulating thymidine kinase, for example, would not have been possible without Bob Livingston's leadership on this study. Bob also was very interested and embraced biologics very often. This is a slide I stole from one he presented at one of our meetings regarding adjuvant chemotherapy in the United States, standard of care, and how he thought we should add trastuzumab if HER2 positive receiving chemotherapy. And of course, we went on and did that. But more importantly, his leadership led to SO800, led by Zena Nale, in which we performed a prospective randomized clinical trial of neoadjuvant chemotherapy with or without bevacizumab, published in Breast Cancer Research about 6 years ago. Breast Cancer Research and Treatment, sorry, about 6 years ago. And as you can see here, overall, this was felt to be a negative study, but I don't think it is. I personally think doing subgroup analysis, with all due respect to Bill Barlow and the dangers of subgroup analysis, the suggestion was that bevacizumab may well be active in patients who had triple negative disease. These are overall and event-free survival data. And you can see here is, although they are not statistically significant, and this may well be play of chance, the suggestion is if we get clever, we could find biomarkers that could bring bevacizumab back out. I'm convinced bevacizumab has activity in breast cancer, but not in all of breast cancer. And if we were clever, we could save this drug and have it again available for our patients and improve, I think, probably overall survival. And I would encourage the leaders now in SWOG to look back at SO800 and see if we can do something. Bob also embraced correlative and biological sciences, now called translational medicine, of course. And I'm not going to walk through all these, but these are 12 papers I could find that we published because of Bob's leadership and his support of our efforts to do translational medicine correlative science studies within the breast cancer studies that we had already accomplished or in prospective studies that he was supportive of. So again, Bob was really a terrific supporter of these things. More importantly, perhaps, in conversations I had with him after I joined the group, his support and mentorship led me to begin to ask, so what? For those who work with me at University of Michigan, you know that I frequently will ask that question after a presentation by a grad student or a fellow who has data that look quite interesting, but really hasn't thought through, how do you translate this? So what? Yeah, nice publications are nice, but how does this help us take better care of patients? Or in other words, nice publications can be like stamp collecting. They make pretty pictures, you can put them in frames. But the real issue is, can you send a letter with this? How does this help the patient do better? And I think we should all be asking ourselves, so what, after every study we do? And that leads into the introduction of tumor biomarker tests in the clinic. 25 or 30 years ago, there was no framework for tumor biomarker research or evaluation. And over the last 15 to 20 years, a number of concepts have evolved that I think are worth knowing about. I think one of the more important ones was published by Steve Teutsch and his group from the EGAP. I can never remember what that stands for. It's part of the CDC. It's an initiative in which they articulated three terms we'd all been thinking about: analytic validity, clinical validity, and clinical utility, which I will discuss in great detail in just a moment. And then ways to test the test. So instead of just hoping you get lucky, the late Dan Sargent, who was at the Mayo Clinic, and Boris Friedman, who's at the National Cancer Institute, both published papers saying, here are multiple prospective trial designs one might consider in order to test a test. And I think they changed everything. We'll talk about that in just a moment. Prospective retrospective studies, I'll get to in a second, and a roadmap for the development of tumor biomarker tests from the Institute of Medicine. I was fortunate to get to publish a paper in the Journal of Clinical Oncology about a year ago. This is a paper that really should have been multi-authored, except that it was my opinion of the things one must consider and how to consider them in order to develop clinical utility of a tumor biomarker test. And I didn't want to embarrass my colleagues by getting multiple opinions onto the same paper. It just got too messy. So I decided to publish it egomaniacally as a single author paper. But I want to thank a variety of people, Lisa McShane, Rich Shilsky, and many others who helped me think this through. What I really said was, what is clinical utility? And this goes back to the EGAP, tumor biomarker test semantics. First of all, analytic validity. Is the assay accurate and reproducible? Because if it's not, it's a lousy assay. I don't care what the biomarker is. If you can't measure it accurately and reproducibly, you don't have a good biomarker. Ki67 is a classic example of that. There's no question that Ki67 is prognostic. There are two major issues, though, is how do you do Ki67 analysis? And IHC is very hard to do and very hard to be reproducible. And then the second question is, if you apply that, what is it you're actually applying to? And I'll talk about that in a second. Clinical validity just means you can take one group and split it into two with p-value less than 0.05 based on some clinical issue or something you think is important. But what really counts is clinical utility. And that is, does the use of the tumor biomarker test improve clinical outcomes? I want to really emphasize that clinical utility is unlikely without clinical validity, but clinical validity does not equal clinical utility. You really got to think about the clinical utility. So these are two sides of the same coin. Analytic validity is essential. Without it, you can't have clinical utility, and clinical utility is essential. And without it, it doesn't matter if you could do the test well. So there are a number of factors that should determine the clinical utility of a tumor biomarker test. And again, I refer you to the paper I published a year ago, but I want to really hit on intended use, and that is, how do you use tumor biomarkers? How can we understand how it will be used for the intended use? Now, this started because I published a paper with Rich Shilsky and Michael Adin and Gary Lyman. Michael is a really talented PhD who was wanting to write about the value of tumor biomarker tests. And she came to me and said, how do you use these things exactly? And I kind of mumbled around and misarticulated. I couldn't really tell her. And she goes, I think what you're telling me is that, first of all, you have to understand the standard of care for the situation in which you're in, for the context of how you're going to use it. And then understanding that standard of care, do you opt in, do you opt out, or do you opt alt? I'm going to talk about that in just a moment here. So opt-in is if the standard of care is to do nothing and the tumor biomarker says you should do something, you opt into whatever you're going to do. I think BRCA1 and BRCA2 are great examples. The average woman down the street finds that having prophylactic mastectomies and oophorectomies is an unacceptable strategy, and I agree, unless they have inherited a deleterious mutation in BRCA1 or BRCA2, and if they have, suddenly those become acceptable. So they opt in order to avoid generating or developing a lethal malignancy. Opt out is where the standard of care is to do something and you decide not to. I believe that endocrine therapy and estrogen receptor is a classic example of opt out. Endocrine therapy, as we all know, especially in the adjuvant setting, reduces the odds of dying of breast cancer by a third to even a half. It's incredibly potent and doesn't have that many side effects. So ideally, we should treat everybody. But we have enormous amounts of data that if your cancer is estrogen receptor negative, it doesn't work. And the Oxford analysis has clearly demonstrated that. And so we readily opt out of giving endocrine therapy to ER negative patients because it doesn't work, even though if it does work, it works really well. Opt-alt is when there are a number of options for a given paradigm, a given context, and you're trying to pick which one. I think non-small cell lung cancer in the metastatic setting is a great example of that, where we now have, I don't know how many, 15 or 20 different possible therapies for metastatic non-small cell lung cancer. I probably overestimated that, but nonetheless, using next-gen sequencing and panel assays, we can pick the one that seems to be best for that patient. We're opt-alt. In other words, a number of options, a number of alternatives, and we pick the one that we think is best for. Does the use of TBT improve clinical outcomes? But really, in other words, do the results of the assay lead to clinical decisions that have been shown with high levels of evidence to improve outcomes? And I really emphasize high levels of evidence and how do we generate those? This is a slide from 25 or now almost 30 years ago, 25 years ago, in which the ASCO put together a tumor biomarker guidelines panel. We'd never done that before, and a number of us kind of sat around and said, how do you do this? And we wrote a separate paper in which we proposed levels of evidence for tumor biomarkers. You can see here, and we really concluded that most tumor biomarker studies were level of evidence 3 and not good enough to change clinical practice. About a decade ago, the Institute of Medicine convened a committee that took that and some other concepts and said, there's a roadmap for developing tumor biomarkers. On your far left is the discovery phase where you've got a good idea. Next to that is you develop a test for the biomarker that's locked down, that has high analytical utility. And then you cross that bright line and you follow one of 3 possible strategies, so-called prospective retrospective study, I'll talk about in a minute, or prospective clinical trial. What's this got to do with us and SWOG? A lot. SWOG has really done a great job of following that. So there are strategies to test the test. I've already talked about Dan Sargent and Boris Friedland's publications. There are really very few such trials that have been performed in the United States or in Europe. For example, in the breast cancer, there are 4 that I know of. 2 of them are from SWOG. SO500, led by Jeff Smiraj, in which we tested whether or not circulating tumor cells after 1 cycle of chemotherapy in metastatic disease could be used to improve outcomes. It didn't. The test did what we thought it would do, but it didn't improve outcomes, so it didn't have clinical utility at that point. But at least we tested whether it would. And SWOG S107, led by Kevin Kalinsky, in which patients with ER-positive, HER2-negative breast cancer were randomly assigned to chemotherapy or not if they had an oncotype BX 21 gene recurrence score of less than 25, and that was just recently published in the New England Journal of Medicine. Is a prospective trial always necessary? No, but it's necessary that you do a retrospective study carefully and with great rigor. And I had the opportunity to publish a paper with Rich Simon two-step hypothesis, and other things that Simon's been famous for. As you know, he was head of the NCI biostatistics for a very long period of time. And Soon Paik, who was the pathologist at the NSABP for a long time too. And we proposed something we called prospective retrospective. Rich actually, Dr. Simon actually coined the term, but what he meant was that you can do a retrospective study, but it needs to be performed prospectively with a formal protocol written out in what you want to do, how you're going to do it, what's the power, the statistics, and it should be using archive specimens from a previously run prospective trial that asks the question that you want to ask. Has SWOG done this? You bet we have. When I first joined SWOG, I had already been dealing with genomics health and the 21 gene recurrence score. The NSVP was about to publish their paper, Soon Paik's paper, regarding the use in B20 and B14 and node negatives. And I talked with Kathy Albane, and she got excited about this. We had tissues because Peter Rabden had collected the tissues. And Kathy worked very closely with GHI to apply the 21 gene recurrence score to 8814, a paper we published in Lancet Oncology, again led by Kathy, that was the basis for S1007, and that is, let's prospectively see whether or not women who have node-positive disease could be categorized as to whether they should or should not get chemotherapy based on 8814, a classic example, in my opinion, of translating a great idea from a prospective retrospective study into a prospective clinical trial. And so this is the kind of concept I think we should all have in mind, which is on our far left, really high levels of evidence, Taylor RX, S1007, SO500. On our far right, pretty crummy levels of evidence, where most published tumor biomarker studies are, that really aren't very helpful, and everything in between here. And I think this is a concept that we should keep in mind as we move forward. And then finally, applying this back into the kinds of things we need to consider to determine clinical utility of a tumor biomarker test. Does it have analytical validity? What's the endpoint you think is important? What's the magnitude of difference between those who are positive and those who are negative that for you as a clinician are good enough to pull the trigger to either opt in or opt out? And what are the levels of evidence to demonstrate to you that you should do that? And ultimately putting all this together as clinical utility. This will be everyone's consensus opinion regarding what the context is, what the marker is, what you as a clinician think you want to do. But if we don't think about this, then it's just people's opinions made up arbitrarily, and that never works. So going back to the IOM roadmap, those of you in SWOG are here. You have the opportunity to do these studies and to do them right. Many of you have heard me say this: a bad tumor marker test is as bad as a bad drug. I'll say it again. You have the opportunity to generate great tumor biomarker tests with high clinical utility. So let's go do it. Let's make Bob proud. So I want to thank all of you as friends and colleagues in SWOG for being friends and for giving me the opportunity to lead the translational medicine part of breast cancer and be involved in the breast cancer committee for now 20 years. I've had a great time. And I want to thank Bob for all his leadership. We dearly miss him. This is a great picture of Bob and his wife, Shirley. Shirley often came to SWOG meetings and came to our annual or our biannual dinners, and we got to know her well. We really missed both of you. Wish you were still around, Bob, to help us. Thanks to all of you. And with that, I look forward to seeing you next fall in person at the SWOG meeting and hopefully at ASCO as well. Thank you.
So unfortunately, Dr. Hayes is on a plane and couldn't join in even for the discussion session. But I thought the acoustics were very good. It sounded like he was in the room. So the next item on our agenda is to review our publications, and I'd like to ask Bill Barlow to walk you through the papers that were published from our group, and thank you all for contributing to these.
Do you want me to use this, or and you can just? Oh, all right. That picture of Bob and Shirley was actually when he was moving from University of Washington to Arizona, And that was his goodbye party hosted at Julie Graylo's house for him. He was also the first chair that I worked with the breast committee, and a terrific guy. So as Dan mentioned, we've published Responder. It came out in mid-December, about a week after the San Antonio meeting. It was very nice to get it out. It took about 6 months at New England Journal, and the results were already a little dated by the time it came out. So the update at San Antonio was actually newer than the publication that came out a week later. We also published S1222. This was a study that was sponsored by AstraZeneca. It stopped after only 37 patients had been accrued because of the change in landscape for that trial, but we were successful in publishing it in Clinical Cancer Research. It had a strong TM component, which I think helped it with publication. An update on S1416, which Priyanka had mentioned earlier and discussed the results from in the symposium, the Translational Medicine Symposium. So we've been working very hard on the publication, the manuscript, and it should be submitted soon. Very interesting work, I think. Corey Spears talked about this at the meeting this morning for the translational meeting, our work with SET 2-3. This was presented at San Antonio in a poster discussion. There is a manuscript, and it's about to go out soon. We're also doing sub-studies from Responder. For example, in the San Antonio presentation, we were looking at distant disease-free interval, which is not in the New England Journal paper. And a lot of questions have been raised about the degree that women took ovarian suppression drugs and the type of chemotherapy. So there were some observational data that we presented at San Antonio that addressed those questions. They will have to have a separate publication about that. It was an oral presentation at San Antonio. Also from Responder, this is a submitted abstract to the ASTRO meeting looking at radiotherapy use in local regional recurrence in that same trial. So hopefully that will be shown at the October meeting of ASTRO. In 221, so this is going to be, I think, a poster discussion at ASCO, looking at vitamin D insufficiency as a risk factor. And so that also was presented this morning in the TM session, some discussion of that. And this is also submitted to ASCO. I'm not quite sure of its status there, but looking at prediction of pathologic complete response to neoadjuvant chemotherapy in SWOG S0800. As Dan mentioned, it was actually a positive trial, despite what he said there. It was a positive trial on the outcome that was PCR. So bevacizumab did have a higher PCR rate than the control arm. He was showing the survival analyses that were kind of marginal there, and we've done a lot of TM work with this trial. And I think that may be the, is that the, okay, there's one more. Also this morning, Translational Medicine, Jaylene Miao presented Accept software, which is an automated counting procedure for CTCs, and did a retrospective analysis of SO500 data. She's presenting it at a Society for Clinical Trials meeting that is in May. That's more about the technical aspects of the actual analysis. It's also it's been submitted for publication in Nature Medicine with Dr. Nanau as the first author of that. And I believe that's the last one. Thank you.
next agenda item is the updates from our patient advocates and then the Translational Research Subcommittee after that. So I'd like to invite Jeannie and Roberta to give us a brief update on their work. Yeah, I think you should come up.
Thank you. You've already heard my new associate has joined us to replace Elda's position as the second advocate. We're a fortunate committee to have two advocates. And so the first thing I want to do is just let Roberta introduce herself. You can see her picture over there, and she'll tell you a little bit about her.
Good afternoon. I'm happy to be here. I'm Roberta Albany, reside in Mount Penn, Pennsylvania. I serve as a patient advocate for the Abington-Ashbourne Cancer Center in Willow Grove, Pennsylvania. And I also serve on very different committee boards with Living Beyond Breast Cancer and other organizations in Pennsylvania. Happy to be here. Thank you.
I told her I'd let her off easy today for her first time out. I'm sure she feels like she's being hit with a tsunami's worth of information in the last few days. I remember when I started about 7 years ago. And I want to send greetings from Elda, who was our second advocate. She misses being a part of things, but as Dr. Pustai said, she's been overwhelmed with her regular work with the Research Advocacy Network. I wanted to share just something on my own. You can read on the slide about the things I'm involved in. Too much, you'll see I don't know how to say no. But one of the projects I've been involved in since inflammatory breast cancer is my primary area of interest and expertise, that I've been working with Dr. JUGS on an IBC scoring system, and we recently had a publication out, a white paper. We'll move into some validation studies on that, but as someone who went for almost 5 months without a diagnosis, myself 20 plus years ago, that's something I'd really like to see happen before I pack it in and retire. So I was excited about that, and it's been great to work with Dr. JUGS on it. And also wanted to let you know about a new resource for brain metastasis and leptomeningeal metastasis. I serve on the Metastatic Breast Cancer Alliance on a subcommittee there dealing with brain metastasis. And we have a new website that we've put together with the help of some physicians and researchers. The website is mbcbrainmets.org. And we're still doing some updates, especially when in the area of clinical trials. This subgroup meets twice a month, and we have speakers present ideas about their research they're doing, and we've helped to form some new studies, and we've been involved in some work with people from various places. But we realized there was such a need for patients to have a place to find information. There just isn't much out there. And Roberta and I also serve on the Patient Advocacy Committee. And in your packet, you should have gotten the field guide and this little guide too, as well, on the team science that focuses on our diversity, equity, and inclusion and criteria. The Patient Advocacy Committee has already gone through Module 6, and we've done our training so that we're prepared as we help with protocol development to make sure that we're really looking at including those aspects into all of our clinical trial work from the very beginning, so that we do a better job of focusing on the appropriate population to reach out to for a trial. There's no point in us just cutting and pasting as we sometimes do as to what we typically do for a study. We need to be looking at the right population and not just excluding men or things like that we've typically done, because that's how we've always done it. And so we we'll do a better job, hopefully, of understanding how things work if we get the right population in there and pay more attention to it. Thank you. And we're here. Use us. Bring us in early and often is the term, I guess we always say, for the trials, but also be prepared. We both have lots of things going on, so you might not get us immediately. Thank you.
It's great. Thank you, Jenny, and Roberta, for the great work that you are doing. These are really, really very good initiatives that you described. Thank you. So I would like to ask next Alistair and Andrew to update you on the Translational Medicine Subcommittee.
Thank you so much, Lars. Hope you don't mind speaking from the back just to keep things moving. Really 3 things to say. First is to remind everybody that every couple of weeks, Bill Barlow, Andy Godwin, myself, and with Alicia in support, have a virtual meeting to try and discuss any new translational medicine projects coming forwards, to harass those who are already doing projects to try and keep momentum going, and generally to try and be supportive of making use of some of the superb resources that we have within SWOG to advance the cause of improved patient care. Second thing I wanted to mention was that we had some excellent talks this morning, and anyone who wants to join the Translational Medicine Working Group, please let Andy, myself, or Alicia know. We'd be very interested in hearing from you. And Andy had organized a fantastic talk on cell-free DNA and one on extracellular vesicles. And really following on from that, we then heard some updates. As you've heard, Yeling Mao gave a great talk on automated counting of CTCs, which is heading for publication, as Bill suggested. 8814 continues to be the trial that yields great dividends, with Corey Spears having presented and now being submitted for publication, something on the SET2/3 index. Is it predictive or prognostic? And then finally, vitamin D deficiency, perhaps particularly in African Americans, may be something that we've got to be very wary of in giving taxanes. And Dan Hertz has been looking at the neuropathic effects of vitamin D deficiency. So there's a lot going on, and we don't intend to take up 10 minutes of your time, but we just wanted to reiterate that we're very keen to hear from anybody who's wanting to do anything in the translational side of things, together with any clinical trials, or to propose new studies or new clinical trials. Thank you, Lars.
Yeah, this is great, Alistair. So what would be the process if somebody in the audience or among the 50+ people who are listening online would want to propose something? They would send you an email?
Please either email myself, Andy Godwin, and or Alicia to let us know that you've got an idea. And what we will try and do is identify a time and a date for one of our once every 2 weeks meetings where we can spend 10, 15, 20, 30 minutes together just hearing what you want to do, and I hope moving through the process to get such a topic going and doing a decent study within SWOG.
So that's great, and I think this is also very timely. And I don't know, Bill, if I can say this, but my understanding from you is that the 1207, the adjuvant everolimus study, we will open the files and we will actually declare whether it worked or not in May this year. So that also means that we really need to start about designing the translational medicines projects around it, because the result will be available before we meet again in Chicago. And also the other large adjuvant trial that completed accrual, the S1418, also is looking for good translational medicine proposals. That study also completed and events are accruing, and maybe in a year or so we will have the results. So I think Alistair and you will be busy, hopefully.
We hope so.
Your email is filling up.
Thank you.
Thank you. So we're going to move on to review the various open SWOG and CTSU trials. And we invited all the champions and people to present, but some people unfortunately can't make it. And I'm incredibly grateful for Priyanka to step in their role. But the first study is actually Andrew Brenner's trial that's ongoing, and Andrew is here to give you an update.
Thank you. So S2007, next slide, This is a study we activated about a year ago for patients with HER2-negative breast cancer brain metastases that are previously treated. For some reason, the slide's not advancing. There we go. So activated in January of last year. To date, we've accrued 6 of 44. We're open or in the process of being open at 141 sites. So it's still kind of slow, and we've been discussing this with the study champions, both for ECOG, ECRN, and for Alliance. And I think there's still a lot of enthusiasm for it, but it's just been a matter of the timing of it and staff crunches. So we're still optimistic that it'll move forward quickly soon. In terms of the AEs, nothing atypical, what you expect for sacituzumab govitecan. So if anybody has considered activating it, please do, and reach out to me if you have any problems.
I'd just like to see by show of hands, out of those that are here, how many of you have the study open?
Thank you.
So about 8 or 9 sites. I think one of the challenges that many sites might be facing is staffing issues, clinical research staffing issues leading to either halting of enrollment or halting of enrollment to studies that are for small subpopulations that are going to enroll, less than 5 patients a year. And that might be common to a lot of us.
That's what I'm hearing from talking to the different investigators. And if I've emailed you and to bug you to find out about the status, thank you for responding. But that's what I'm hearing right now, is it's just taking longer than usual due to inadequate staffing and the crunch that everybody's experiencing within their clinical trials offices.
So one of the things that, and not every institution might have it, but many institutions might have special categories for highly unmet need. Progressive brain metastasis is a category that often many of us don't really have multitudes of trials to choose from. This might be the only one. So even if it's not going to accrue 10 patients a year, it is still worth having it available for those few patients that may benefit from it because they otherwise will not have another option. Thank you, Andrew. So next, we're going to move to 1706, and Dr. Chalasani, if she's online, she'll present it. Otherwise, I will take over. I don't know if she's online. Dr. Jaxie couldn't be here.
So I think the
Another commitment.
participants online can unmute themselves, right?
Yes, they can. And Pavani might not be. I think she had another conflict that she let me out. So 1706 is a trial that Elder referred to. It's a randomized phase 2 study of olaparib administered concurrently with radiation therapy or radiation therapy alone in patients with inflammatory breast cancer. It's a 300-patient study with IDFS as the primary endpoint. Currently has 161 sites approved, and as of April 1st, 100 of 300 patients have been enrolled. The anticipated enrollment was 5 per month, and you can see this figure that Bill always generates for us for these meetings. And we see that many months we're in that 3 to 4 range, but few months the enrollment does dip down. And again, it's for a unique patient population, and primarily open at bigger academic sites. And we really appreciate the advocacy committee helping us keep the awareness up for the study. Any questions about the study? So the next one is a CCTG MA13. Again, Reshma is our champion. Reshma Jaxie is a champion, and she couldn't be here, so I'll just provide a brief update. It is a trial for patients with 1 to 3 positive nodes and randomizes them to no regional RT versus regional RT based on oncotype recurrence score of less than 18. So the score has to be less than 18 to enroll in the study. I think the studies had some changes in the eligibility and time requirements from various steps to allow more patients to be eligible. macro or micromets are now permitted if it's a T3 disease. Patients with sentinel lymph node biopsy alone must have 1 to 2 positive axillary nodes, and those with additional micromets or ITCs are eligible. There's some mild changes in the age criteria and extension of time period allowed from surgery to enroll, extending to 16 weeks from 12 weeks. So hopefully all of these measures will allow more patients to be eligible to participate in the study. The next one is COMPASS HER2 trials. The first up is the COMPASS HER2 PCR. Dr. Tang is our champion for that. He was going to present virtually till he realized that he misread his calendar and he had to be at his child's school for some sort of a presentation, which obviously is a lot more important. So I'm stepping in for him. This trial is enrolling very well, and it should complete enrollment fairly quickly, I think sometime this year. It's a single-arm study looking at THP for 4 cycles and using PCR to guide further treatment. I think we have the enrollment number up there. 1,100 out of 1,250 have been enrolled, and at this rate, it should complete enrollment this year. So thank you to everyone for supporting this study. The next we have COMPASS HER2 residual disease, and Dr. Murthy is our champion, and I believe she is online. Rashmi, if you want to unmute yourself, let's see.
Hi there, can you hear me?
Yes, we can.
Okay, perfect. Hi, everyone. So yeah, for the COMPASS HER2 RD trial, so this just to remind you, it's for patients with residual disease after neoadjuvant chemotherapy for HER2-positive breast cancer. And these patients are randomized in a 1-to-1 fashion to T-DM1 times 14 cycles or T-DM1 plus tucatinib for 14 cycles. Briefly to remind you of the eligibility, if they are ER negative, then they can have any amount of residual disease. If they're ER positive, then it has to be lymph node positive, although with amendment number 1, they did change that. For low ER positive patients, they could just have in breast residual disease. Next slide. So this trial was activated last January. And thus far, 125 out of over 1,000 patients have been enrolled. And they've activated 898 sites in the US to date, and it also opened in Canada in March of 2022. With Amendment 1, I mentioned the point about the weekly ER positive patients being allowed even in the setting of node negative disease. And then also for patients who received de-escalated therapy off of the parent trial EA1181, they are allowed to receive, if they receive greater than or more 2 cycles of treatment pre-enrollment. And then finally, this spring, in Amendment 2, there was a requirement for AL&D in patients with micro and macrometastatic disease removed. Specifically, if patients have isolated tumor cells in the setting of residual breast disease, then they are required to have either AL&D or planned nodal irradiation, so level 1 to axilla. And if patients have micrometastatic nodal disease, planned nodal irradiation is required.
Thank you, Dr. Murthy. So this is a relatively new study, but a very well-designed study, and our patients with residual disease after preoperative anti-HER2 therapy and chemotherapy are perfect candidates for this study. So next we have BR004, and Dr. Cobain is our champion for this.
Yes, thanks very much. So this is just the amended schema for the BR004 study, which is a trial in first-line metastatic HER2-positive breast cancer. You can see the stratification factors that are listed there. Should also mention that measurable disease is a requirement for enrollment on this study. And essentially, patients are randomized to what our current standard of care is for first-line systemic therapy, which would be a taxane plus trastuzumab and pertuzumab, or the experimental arm, which is that same THP backbone in addition to immunotherapy with atezolizumab. And you can see that now really the main focus of the amendment was to allow either for paclitaxel weekly or docetaxel administered once every 3 weeks as the chemotherapy. Next slide. So the status as of really just a few weeks ago, you can see that the number of approved sites is 596 total. The number of patients screened has been 279, and so far the accrual is at 182 out of a goal of 600. So we're really only about a third of the way along with this trial. It's been slow to accrue for a variety of reasons, but I think one of the main, perhaps, that this is a smaller population, seeing patients with metastatic HER2-positive breast cancer, and that's largely a testament to the effectiveness of our neoadjuvant and adjuvant systemic therapies for early stage disease. The current accrual is about 8 patients per month, which is about 20% of what was projected. And then I don't have the figures updated as of March 2022, but as of November of 21, actually about 78% of patients enrolled had de novo metastatic disease. So this was their very first treatment. And we had a limited number of patients so far, just 2 with brain metastases. So the amendment that was activated in July of 2021, as I mentioned, docetaxel was allowed in addition to weekly paclitaxel. It also allowed you to enter eligible patients based on the local HER2 status. We're hoping that reduced the delay in getting patients enrolled. It previously required central confirmation of HER2. It's also allowed to administer the loading doses of trastuzumab and pertuzumab and the first 3 weeks of chemotherapy while awaiting the confirmation of the HER2 status centrally and the determination of PD-L1, which is a stratification factor prior to randomization. Also, the restrictions, I won't go through all the details, but have been a little bit loosened in terms of those patients who have brain metastases. And use of Phesgo is not currently permitted at present. So just wanted to mention that since a lot of patients on standard of care would be receiving Phesgo.
Thank you. Thank you, Erin. So hopefully all of these changes will help with the enrollment. The good thing is we're seeing less and less of these patients in our clinic, and that might primarily be the reason underlying the slow accrual. It is interesting to see that almost 80% of these patients had de novo metastatic disease. And in the breast committee meeting yesterday, we did discuss and brainstormed about developing clinical trials for patients with limited volume de novo metastatic disease, since these patients are now quite overrepresented in our first-line trials. They may not make up for a large proportion of total breast cancer diagnosis, but clearly they're receiving their first-line therapy along with other patients who are developing metastatic disease after having had initial curative therapy. So the next one is Alliance 011202, and I'm going to skip that. We'll come back to it. Dr. Maman is at the Melanoma Committee, and he was going to be here after 4:30. So we'll move to NRG BR007, and Dr. Shumway is our champion for the study. He's not here in person. I'm hoping he's.
Yes, can you hear me?
Yes, we can.
Great,
Hi, Dean.
thank you. Hi, thanks so much. Yes, so this is a clinical trial that's looking at omitting radiation in the most favorable patients. Many of the historical studies that looked at omitting radiation had very heterogeneous inclusion criteria. And the hypothesis of this study is that we now have the ability to identify patients who are much lower risk. So this is a group of patients who are aged 50 to 70 and have a tumor that's less than 2 centimeters with an oncotype recurrence score of less than 18. And they're randomized between standard of care therapy with radiation and endocrine therapy versus just endocrine therapy alone, so with omission of radiation. And this is a study that opened in June of 2021. There are many sites that are still in the process of getting it opened. At present, we're at about 60% of our accrual goal, and it could use some support if you felt like you could open it at your institution. It's a study where we're asking patients to accept a randomization to not receive radiation. And we found that it's very important for patients to hear about the study across the multidisciplinary team, so that they're getting the same message from the entire team. Hope to have another update with more sites open and increased accrual next time we meet.
So I was just also going to ask by show of hands to see how many of you have this study open at your site. So we see about 6 or 7 hands up, Dean. Since it's a relatively newer study for us, and we have other radiation oncologists present here in the room, I'd like to hear about your experience regarding this study and institutional protocols for either IORT or other partial radiation plans that you might have. And because these are common challenges for many sites, and it's useful for us to hear how different sites are handling this as they're trying to support more than one study for somewhat similar patient population. Do you want to come to the microphone so those that are virtually can hear? Thank you.
So we actually don't have it open because we do offer IORT, and this is the exact population, and you can get salvage radiation later. If you have a recurrence in the future, the nice thing with that particular modality is salvage is there, and it's done while you're in the operating room. So it's very hard to have this as a competing option. So I'm sure it's similar at other institutions with it. The other problem is 5 fractions is incredibly common now. So if patients are concerned that they may not stay on hormonal therapy, it's very easy to take 1 week of radiation.
Yes.
So those are our issues.
Yes. And we have another comment?
Yeah, I'm not a rad onc, but we have this open at our community site in Oregon, and I've actually put, I think, at least 2 people on. I have a few people in the hopper. My rad onc partners like the idea that we don't do IORT. I think if you get an engaged, group of people who aren't maybe at such a specialized center, you can have success. So I agree. I definitely know they talk to patients about 5 fractions and less, but the few people I've found have been really interested in just avoiding radiation altogether if they're randomized to that. Yeah.
So is might be a better fit for certain practices.
Right. This is great for NCORE sites,
yes.
Especially if you find your radiation partners, and once you all talk about it in your tumor board, they just start thinking of it, and it's nice to have 1 or 2 protocols in your head. It's easier to enroll. So.
And for places where patients are traveling quite a bit of distance to have radiation.
Yeah, that's a lot of the people we serve.
Yes, so, and because for some of them, that actually the decision between the mastectomy and a lumpectomy, not what they need is, can I get to radiation? I can't get to radiation, so I'm going to choose a higher level of surgery. So, Dean, I want to give you an opportunity to respond back to some of the other challenges with competing studies. We do have one more question before that, though.
Yeah, no, it wasn't a question on RADONC. The points were made already that we have intrabeam at our institution and the 5 fractions. So many women do have trouble staying on the hormone medications. And so they are worried because they haven't yet been on it. And in fact, our med-oncs don't really like to start the hormone medications until after the radiation's done, because worrying the patient will go off of it if they blame it for a side effect. So it's a very complicated issue if the patient gets no adjuvant treatment versus one that they can't sort of take away once they've had it.
So, Dean, do you have any pointers on how to frame our responses to patients or how to counsel them for these very legitimate issues and concerns?
Yeah, thank you. I appreciate the discussion very much. I think that those are very reasonable concerns that the patients raise with the difficulty of completing a 5-year course of endocrine therapy. I do think that there are many patients who are highly motivated to avoid radiation. And I think maybe just emphasizing the very low risk in this, small tumor, ER positive, low oncotype score, that we think will be a remarkably low risk and that they'll do quite well even without radiation. But I certainly do think that it does need to be a study that's supported by the multidisciplinary team. And I'll bring these comments back to the study team as well.
Thank you. So we'll move back to 11202. I see Dr. Marmon just walked in.
This is a very anticlimactic update for A011202, which is that currently, as it has been for the last several months, accrual is on hold. The number of eligible patients of the total, once accrual had been met, was inadequate. So there's a amendment that's going through CTAP right now, and hopefully it'll be opened again in the very, very near future. So Dr. Bowie was encouraging us to continue to hold tight and was looking forward to reactivating the study. Again, just briefly the scheme, I know we've seen this many times. These are individuals who are pre-registered, undergo a sentinel lymph node, have neoadjuvant chemotherapy, and then have a clinically negative axilla after neoadjuvant therapy, after having a positive lymph node initially, go to the operating room and are found to have a positive sentinel lymph node, and either have axillary dissection or just a sentinel lymph node biopsy and then radiation afterwards. So they're randomized in the operating room. So hopefully we'll hear about it opening again, and I'll keep everyone posted.
Do you have any news or insight into by how much the accrual is going to need to be extended?
Yeah, I don't actually. Okay. Dr. Bowie didn't say. I think she's still in negotiation as to that number.
Absolutely. Right. I think we have a comment in the back.
Yes, sorry. The radiation arm on this has instructions that are very hard to meet. And for example, if a patient has a scar that goes all the way around her back because they wanted to avoid the ear that forms if they don't on a mastectomy, we have to radiate that whole scar to the full dose. And to do that, we'd have to radiate a lot of lung. And so we don't like the instructions on that. I think they were copied from another protocol that got amended because of some of the same issues. So it might be good to just update those and let people know, because every time I have to do a case, I complain to TJ, and he always convinces me to find some way around it, and so I do. But it's a problem.
Since there's an amendment in the works, maybe we can incorporate that feedback. Thank you.
Thank you, Dr. Marmon. The next one we have is SWOG 1501. Dr. Floyd, I believe he's also attending virtually.
Yeah, I'm on. Can you hear me?
Yes.
All right, so this is a, you can go to the next slide if you want. This is a randomized trial looking at carvedilol versus no intervention to reduce the risk of cardiac dysfunction in metastatic HER2 positive patients receiving trastuzumab-based HER2 targeted therapy. It was a 3-arm study. The non-randomized arm has been closed to accrual. We met our accrual. And so now the two randomized arms are open still. Patients are followed for 2 years with echoes every 3 months. This is sort of a slide of accrual. And as you can tell, there's sites that have had very robust accrual. And we want to say thanks to the high accruing sites. We are up to 265 patients accrued. As I said, the observation arm has closed. We've recently made some amendments allowing up to 4 lines of therapy. Brain mets are allowed. We've incorporated the newer anti-HER2 drugs, biosimilars, et cetera. We've also met recently. I presented last month at the Alliance Symptom Control Committee. Dr. Ruddy is now our champion in Alliance, so we're hoping to get additional alliance studies on board. I've reached out to the breast committee to present at Alliance Breast Committee, and I'm going to reach out to the other cooperative groups and try to engage the other cooperative groups to get additional sites open and pick up on accrual. We have done our baseline neuregulin assay. That's one of our biomarkers, and have started doing that analysis. We are looking for sites, and so check and see if this is open at your institution. If it's not, we need more sites. And as you can tell, patients, the patients are out there, and sites can have robust accrual. And I guess I would just like to remind people as well that patients do not need to be starting a line of therapy. So they have to be within the first 4 lines of therapy, but whatever line they're on, there's no limit to the number of treatments they can have. So a simple screening strategy is just to have your pharmacy run a list of patients on trastuzumab or related drugs with breast cancer and screen those patients. And you don't have to wait for them to progress. They can be put on any time in their treatment cycle. So thanks for your time. Appreciate it. Reach out to us via email and open it at your site if you don't have it open.
Thank you. And I just realized that the slide for BR003 is missing from the deck. So we do want to discuss that study. Dr. McCool's on with us virtually. And I'm sorry, Sam, I don't know what happened to the slide. It is not here, but you have some good news to share with us.
Yes, thank you, Priyanka. The trial, as you remember, it's an NRG BR003. It's a randomized trial that enrolled patients with node positive or high-risk node negative, triple negative breast cancer patients. They were randomized 1-to-1 to dose-dense AC for 4 cycles, followed by 12 weekly treatments with paclitaxel, or the same treatment with 3 weekly carboplatin that begins with the weekly paclitaxel. The trial was activated in June 2015, and the endpoint, the primary endpoint was invasive disease-free survival, and the secondary endpoint, overall survival, toxicity, and other correlative studies were designed around it. The good news that I have, actually, this trial had a lot of difficulties in the beginning to accrue patients. However, now we reached the total accrual of 767 out of 782. So we are 15 patients away from the finish line. And I truly believe that by probably the anniversary of the opening of the trial, we will have finished accrual. This is a very important trial, as you all know, that we still at this point don't know exactly which group of patients carboplatin will offer help. We know that this is a toxic drug. We have seen at San Antonio that carboplatin doesn't offer benefit, for example, to some patients with basal cell triple negative breast cancer. So there will be a lot of, in my opinion, a lot of advancement of this question at the conclusion and the maturity of this trial. So please, for those of you who have the trial open, we are very close, and I think we count on all of you, and thank you for all of those who contributed patients to enroll on this trial. I want to here highlight the fact that SWOG have contributed 90 patients, and that was the second most important contributions among all the other cooperative groups. Thank you.
I believe there was an email today that the trial enrollment, putting out a date a week or so out, but beyond which we won't be able to screen. So it's very, very close, right. As you said.
Yeah.
And I apologize, I don't know what happened to your slides. So,
that's fine. I hope that people were able to listen to the message, and that's the most important thing.
This is a test of multitasking. So I have a slide up for 1501, but we're discussing a different study. So, okay, thank you. We'll move on. The next study up is 1703, and I think, Melissa couldn't be here, so I'm going to present that on her behalf. keep a list of who all I'm going to be today in one day. So this is a randomized non-inferiority trial comparing overall survival in patients monitored with serum tumor marker-directed disease monitoring versus usual care in patients with metastatic hormone receptor-positive breast cancer, chaired by Dr. Accordino and Hirschman. The trial has champions now from other cooperative group. Dr. Javeri is the new champion from Alliance, and we should have a champion from ECOG Akron as well. So these are the enrollment numbers for the study. As of a couple of weeks ago, the trial has enrolled 214 out of 1,056 patients on the step 2. Dawn, you're here. Do you want to say anything about the study?
I would only say that the study's extremely, there have been a lot of modifications to this study to make it very easy to enroll. So if you had it open and it was challenging in the past, you should relook at it, because there are about 20 different modifications that were made, both in terms of the time of accrual. You only need one elevated tumor marker, a whole slew of things that people provided feedback for first. Now it's much, much easier. So I would say any barriers that are in the past, to relook at it. I don't know if any of the slides show there were a gazillion changes, but people should relook at it because it's now quite easy.
And there's a question on chat. Banu Symington asks, do you have to be an NCORE site to participate or no? So
No.
It's not an NCORE specific study.
No. Nor is it under CCD. So she could go ahead.
Go ahead, open it, yeah. Thank you. Okay. So next we have EA 1183, and Dr. Linden's here to give us the update.
This trial is going quite well and rapidly. It's completed. It's halfway through to completing accrual, and SWOG, I believe, is about half the accrual. So thank you, everyone. This is Jen Specht and Heather J. Seen's trial looking at bone-dominant metastatic breast cancer. I think this is a very important trial because these patients were excluded from every other trial that we're basing all our decisions on. So this is about how we can get a handle on lytic lesions with FDG PET. So Jen asked me to encourage people to sign up for the optional blood collection so we can look at those important markers that Dr. Hayes emphasized. This is nicely funded by a companion oral one, which does involve some extra imaging, but that's not mandatory at this time, and I think we're feeling pretty good about this. I think there may be one other slide there.
Yes.
I think this is exciting. It's a little awkward having put a bunch of people on this to have the research coordinator chasing you around about the timing of your scans, but I think this is going to be a very useful data set for us, and thanks to SWOG for the participation.
Thank you. So it looks like the study has good support and is accruing very well on target.
Yes, but there is still time for other sites to join if they'd like to.
So does this require a special PET or?
It's just FDG. I think you do have to be Akron certified, which means you have to be able to pass a few tests, but it's not a huge requirement. It's really just some timing. I don't think you have to have measured a phantom.
Okay. So the next is TMIS, and Dr. Arun couldn't be here to give an update in person. So this is a randomized clinical trial of screening mammography. It's, like all screening studies, it's a pretty large study comparing breast tomosynthesis to digital mammography. And annual screening at entry in 1, 2, 3, and 4 years after entry, and biannual after that. The accrual is at almost 66,000. Let's see if he has another slide about accrual. Okay, that was the last slide. So thank you for those that are supporting the TMIF study. And I think it's also one of those studies that is perhaps doing better at some of the NCORE sites that are offering both types of imaging. There are many sites that are doing just tomo, so they don't have enough volumes of regular mammography to offer the study. So this concludes our review of open CTSU trials, and we'll move on to hearing some updates on concepts that you all have heard before. The first one in the queue is a SWOG 2202 or DETECT-01, which will be presented by Dr. Bardia. I'm hoping he's on. Or Dr. Kalinsky on behalf of the team.
Yeah, I'm happy to present. Can you hear me?
Yes, we can.
Great. So we would like just go to the next slide.
Next slide.
So we've talked about this study, which is now approved via SWOG triage and CTEP as well, which is looking at ctDNA with the hypothesis being that ctDNA can identify molecular relapse before it becomes clinically apparent. Next slide. So with feedback that we received from CTEP, we have changed the design of the study. The population is similar in that this is including patients with stage 2 or 3 triple negative breast cancer with residual disease after neoadjuvant chemo. And we would be collecting for those patients who are going to be receiving radiation therapy, ctDNA during that time. And we would ultimately be expecting that of the 300 patients or so that we would be collecting ctDNA, about 94 would be randomized if they do not, we would have already determined they don't have clinical radiographic evidence of metastatic disease from scans that they would have recently had, and they would be randomized 1-to-1 to capecitabine versus sacituzumab govitecan for 6 months. And you can see that we're collecting ctDNA every 3 months, and then we also will be performing CAT scans that will be covered at 12 months and 24 months, with the primary endpoint being ctDNA clearance. And after 2 years, both of the arms would revert to standard of care follow-up. And you can see some of the secondary endpoints included here as well, such as relapse-free survival at 2 years. Next slide. These are the statistics. And as I mentioned, it's 94 patients. And we did incorporate some stopping rules per the recommendations that we received from CTEP. Next slide. And there is an active TM specimen collection where we'll be collecting tubes in addition to the ctDNA that we will have sent to Natera. And we are requiring, of course, that blood and tissue will be sent directly to Natera for ctDNA analysis prior to randomization. And the additional blood and tissue will be stored first at the SWOG repository. Next slide. So just as an update, as I mentioned, this has been triage approved by CTEP. We have a protocol first draft, and we are anticipating PRC review in May. Next slide.
I think that was the last one, but I do want to ask you, Kevin, can you remind our audience how adjuvant pembrolizumab will be handled in the study?
Right, so currently we have it so that patients would be receiving their capecitabine or their sacituzumab govitecan, and then patients would then, upon completion, have the option for continuing their pembrolizumab if deemed standard of care per their treating physician.
Okay, so
Kevin, also, if you could just sort of clarify that the eligibility is a one-time ctDNA positivity after surgery, right?
That's correct.
And can that test take place before radiation, after radiation, or that does not matter?
It does not matter. It does need to occur before a patient would theoretically start capecitabine before randomization, but it can happen at any time between surgery and the time that they would eventually start their adjuvant systemic treatment, chemotherapy.
Which is the 12-week window, so given that it may take 3 weeks for the test to come back, the earlier the better.
Correct.
From that time period. So we'll do a poll question on this, but before we get to the poll question, I just want to make sure that everybody understands how this polling will work. So if you would all pay attention to this slide and instructions. So there's 2 ways to join the poll, and they're exactly the same whether you're here in person or you're attending virtually. So I encourage all our virtual attendees to participate in the poll so we really can see what your level of enthusiasm for this upcoming study is. You can join it by web or by text. If you use the text to the number 22333 and text the word Courtneyville 940, and that will join you in the poll, and then you just see the questions and provide your answers after picking the option. So we'll just do a test question before we go into an actual question. So this is the test question. How are you planning to attend ASCO? And we see many of you are responding.
Very few of them are responding.
So more and more are figuring it out. We can rerun the test question, just to give all of you more time to figure this out and join this. Can we rerun this test question?
So, I think we probably have about 40 people in the room and another 50 some online, and we only have 15 responses.
It's not letting you respond. Let's see. Yep, I think now it's working, right? Yep, we see the number going up just a little bit. Going down, somebody retracted the response.
Yeah, so this is basically just to make sure that the system works. Although we are interested to see you in ASCO, but the interesting questions that we want to hear your feedback will come after this.
Okay, so about 28 of you could join the poll. So we'll move on to the actual question related to the study. The DETECT 01 or 2202, will you open this study at your institution? And the options are yes, no, due to competing studies, no, I don't like the trial design, no, I will not have many eligible patients for this, or maybe I'm still on the fence about what to do with the study. And we may need to extend the timer on this again, if you don't mind. So we've about 30 responses, and about 30–40% will open the study as is, and we have a good number of investigators who are on the fence at maybe. Few have competing studies, which can always be the case. So for those that are at maybe, we'd like to hear from you, because this is the time, because the protocol is being developed, and so if there are any major concerns that can be addressed up front, we would like to keep them under consideration. If you're virtual, you can unmute and speak. If you're here, please come to the microphone.
So I see Kathy raised her hand. virtually.
Kathy, you should be able to unmute yourself.
yourself.
You should be able to unmute
I
yourself.
am.
Yes.
And then I got remuted by someone. Okay. Kevin, what is the going percentage of patients in this eligibility space who will be ctDNA positive by this assay?
So we're estimating that it's about a third, and then we're estimating that, you know, maybe about 10% or so may not proceed to randomization, but we're estimating that it's about a third.
Just to, as a background to this, so this design came about because of the C-TRACK trial results that many of you have seen at the San Antonio breast cancer meeting. So that study was a similar design, and they tried to test pembrolizumab in that context. And the remarkable finding was that the largest number of positive tests was actually seen at the beginning, right after surgery. And it was also seen by the I-SPY study. So the yield is really the highest at the first testing. And during the follow-up, when people get tested like 3, 6, 7 months after their surgery, at the time, they actually had metastatic disease, overtly frank metastatic disease. So the premise that the ctDNA identified patients at a preclinical stage, an undetectable micrometastatic stage, did not hold up in the C-TRACK study. Folks who had ctDNA positivity had actual metastatic disease, which was asymptomatic but detectable on scans. And that led to this design that we actually restrict the eligibility to right after surgery being ctDNA positive. And that, as Kevin pointed out, based on the existing data, which is somewhat thin, but it does suggest around 30% of the people have that. And if they have that, they do really poorly. So that's the poorest of the poor prognostic group among the residual disease patients.
We have another question from Dr. McCool.
Yes. You know, all of us have seen the data about the effect of capecitabine on the basal-like triple-negative after neoadjuvant chemotherapy or even adjuvant, based on multitude of trials that were presented over the last 2-3 years. And I wonder here, in our institution, we took notice of that and we started checking the patients either with PAM50 or CK56 or EGFR expression to see if they have the basal-like subtype or not. And our investigators have now a strong reluctance to give them capecitabine because they think, why do I have to give somebody a treatment that doesn't help? So I don't know how the study would be able to address this question. At least having that as a stratification factor would entice people to enroll on it, because we will be able to put this question to rest, perhaps. The numbers are small and probably we would not be able to reach this conclusion, but at least somebody would say, okay, you know, we're trying to understand the biology of these residual tumors.
So thank you for bringing this up. And as you mentioned in the ECOG-ACRIN study, they saw for those basal-like tumors that there was limited response to capecitabine. I just want to mention in this study that we will be encouraging tissue collection for future correlative studies. And I think the point about, you know, potentially having something as a stratification factor, as you highlighted in a study which is less than 100 patients in terms of how many would be randomized, that may be somewhat difficult and limited to do, but I think there's definitely an opportunity for us to retrospectively go back and evaluate not only tumor tissue, but also other potential prognostic and maybe predictive blood markers as well.
So I would just follow that with the comments. So these are patients with residual disease and ctDNA positivity. Would providers still not be offering capecitabine to this highest risk population? I mean, most of us would, if there was not a trial, we would be offering them something. And the standard of care, in the realm of standard of care, that is the only drug right now. We have a comment here, Aaron, yes.
Yeah, so two questions. Hi, Kevin, it's Erin Cobain. I'm sure this has been discussed before, but just curious about your thoughts. One just concern I would have about accrual to the study from the patient perspective is the notion that their adjuvant systemic therapy is going to be lengthened quite a bit by participation in this study. Totally understand the rationale of not administering particularly sacituzumab and pembro concurrently, but that I think just might pose a challenge. So I'm curious about your thoughts on that. And then I guess the second question that I have is just, are patients with a germline BRCA mutation ineligible for this study because of, our standard would be to give them adjuvant olaparib? I'm assuming that's the case, but just wanted your thoughts on that too.
Yeah, I'll talk about the second question first. Yes, we are only including patients who are BRCA wild type. And for the first question, it is a very good point. We've debated about when we could theoretically initiate pembrolizumab, and there have been multiple conversations about whether we should include that during the time where patients are taking CAPE or SASI. And the ultimate feeling was one that it could be confounding. And there are other studies that are looking at specifically the combination of an ADC and checkpoint inhibitor. So that's why we landed where we were. And then the other element, which I thought you were going to go as well, but just to also add, we've had conversations with Lynn as well, just about the incorporation of patient-reported outcomes, just given this updated design.
And then in terms of, the concurrent administration of CAPE and pembro, I know, many of us might be doing it in clinic. But to recommend that as standard on a CTAP-supported study was also a bit of a challenge because that's not something that's in any of our pathways. The safety data is from a small metastatic study, and so those were some of the other considerations that went into the final design.
So, Erin, the chemotherapy component of the treatment is the same, right? So you just substitute the capecitabine with the sacituzumab. And the value of the adjuvant pembrolizumab in that setting is completely unknown, right? I mean, it just happens to be that the trial was designed, Keynote 522 was designed with an extended adjuvant pembrolizumab phase. But whether that contributed anything to the outcome beyond what was accomplished in the neoadjuvant setting is really unknown. So if you're one of those that does your CAPE and pembro sequentially, then it wouldn't lengthen the duration. But if you've migrated to doing it together and have been able to get insurance coverage for it, which is starting to become a bit of a challenge, then it wouldn't technically lengthen the duration for the patient. Any other comments for those that were in the maybe category about the study design, drugs, ctDNA testing? If not, we'll move on to our next concept update, which will be given by Dr. Cobain.
Thanks very much. So this is another concept that has been recently approved by the Breast Cancer Steering Committee, looking at neoadjuvant immunotherapy plus chemotherapy in a subset of patients with estrogen receptor positive, HER2 negative breast cancer that are mammaprint ultra high risk, designated as MP2. Next slide. So we really wish to test if the addition of durvalumab, a PD-1 inhibitor, to standard of care neoadjuvant chemotherapy may improve invasive disease-free survival in this specific subset, genomic ultra-high risk mammaprint MP2 hormone receptor positive breast cancer. And we have secondary endpoints as well, which we think will be very important and informative, including pathologic complete response rate. There is a lot of work going into designing quality of life and PRO studies led by Dr. Irene Kang, as well as some exploratory biomarker assessments, which we won't talk a whole lot about today, but is being led by Dr. Justin Balco. So this is the schema, and essentially it's a population of hormone receptor positive, HER2 negative early breast cancer patients who would otherwise be eligible for chemotherapy per standard of care. Ultimately, the goal is to accrue approximately 960 patients to this study. And there is a pre-screening step, which is a mammaprint assay, which is provided free within the context of this trial. And if patients are MP2, that mammaprint ultra high risk, they would be randomly assigned to either standard neoadjuvant chemotherapy, which is weekly paclitaxel followed by dose-dense AC, versus that same chemotherapy backbone plus durvalumab, the PD-1 inhibitor. Patients would then go on to surgery, and then they would have standard of care adjuvant therapy to include radiation if indicated, endocrine therapy, CDK4/6 inhibitor therapy with abemaciclib if indicated, and just wish to mention that there's no plan for any maintenance immunotherapy component in this study. Patients will be stratified by a few factors, so age, less than 50 versus greater than or equal to 50, their nodal status at presentation, as well as stratified by intended abemaciclib use. Been a lot of just discussion around that, and we do anticipate that there's potentially going to be more abemaciclib use in the control arm, particularly if durvalumab improves rates of pathologic complete response as we anticipate. And as I mentioned, the primary outcome will be invasive disease-free survival. Secondary will be path-CR, and then I've listed our more exploratory biomarkers there as well. So this is the key eligibility criteria. Essentially, again, it's just a population of patients, hormone receptor positive, HER2 negative, for which chemotherapy would be indicated as standard of care. So we've sort of listed there all of those criteria, including, those that have high oncotype, premenopausal women that have known lymph node involvement at presentation. But just wish to emphasize, in order to qualify, right, for randomization in this study, we must have the mammaprint assay. So one of the things we've just talked about, with perhaps, moving things along as quickly as possible as you're aiming to accrue patients to this trial. When we know we have a hormone receptor positive, HER2 negative early stage breast cancer patient, it may sort of behoove us to get the mammaprint early on from that core biopsy. So they must be mammaprint MP2 status, as I mentioned. Next slide. So, the MP2 subtype is estimated to be approximately 10 to 15% of all early stage ER-positive breast cancer. So we estimate that's approximately 13,000 patients per year diagnosed. And among mammaprint high risk, which is the normal sort of indicator that would come back on the report, this MP2 is sort of something that's unique to this trial that would be designated. About 50% of the mammaprint high risk are in this MP2 category. There's approximately an 88% concordance between Oncotype DX recurrent score greater than or equal to 30 and mammaprint high risk status. So just if these patients have Oncotype first, gives you an idea of what proportion are maybe likely to ultimately qualify for randomization. And there's going to be a data safety and monitoring committee that will review the MP2 detection rate just to determine if any changes in the screening strategy are needed. The mammaprint, as we mentioned, can be ordered completely free within the context of this study to determine chemotherapy eligibility. We also anticipate that the mammaprint result would turn around in approximately 7 to 14 days. So if it's ordered, from the get-go, once the diagnosis is established, we think this would be in a very reasonable time frame prior to the start of neoadjuvant chemotherapy. It can be ordered in parallel with Oncotype DX recurrent score. The Oncotype would be charged to insurance, and then of course the mammaprint would be free, just if that's sort of institutional practice, but you could also just order the mammaprint alone from the get-go. Next slide. So the goal accrual will be approximately 25 patients per month. This is the estimate actually of patients that would be randomized per month. So this is specifically the MP2 subgroup. And just for reference, the S1418 study accrued about that number after its initial ramp up. And we estimate that the proportion of patients eligible for this trial with that MP2 status is similar to the number of triple-negative patients with greater than 1 centimeter of residual disease, which was the population for S1418. And then the I-SPY and industry trials we know of that are potentially competing. The I-SPY trial, as we know, really has been running in parallel with several other large neoadjuvant industry trials, and all have been able to actually stay on track and maintain accrual goals. And then with regards to the industry studies that are listed there, I think that both of these are going to be wrapping up in the near future. And so by the time we get this study actually open and up and running, we don't anticipate that there'll actually be major competition with those studies listed. So just in summary, this is a phase 3 trial prospectively testing the hypothesis that's, and I think my one slide was missing at the beginning, so I'm sorry if I didn't mention this, but
this one?
No, it was the rationale for the study in the first place and why we're doing, basically using the MP2 as randomization. But that slide basically just indicated that in the retrospective biomarker analyses from the I-SPY 2 trial, we learned that those patients who had this MammaPrint ultra-high risk status actually appeared to have marked improvements in rates of pathologic complete response when immunotherapy was added to standard chemotherapy. So that's really the whole background and rationale for this study. And so this is basically testing that hypothesis in a prospective randomized trial. At present, there really isn't an ongoing trial in the NCTN for high-risk ER-positive early-stage breast cancer patients, so this really fills that void. It could also address whether or not pathologic complete response leads to increased invasive disease-free survival in selected ER-positive breast cancers. And this would be the only neoadjuvant immunotherapy study that does not include a maintenance immunotherapy component. And then the last slide is just our progress to date. So we were approved by SWOG Triage in September of 2021, submitted to the NCI for Breast Cancer Steering Committee review in December, ultimately got approved in February of 2022, and the final approval is just pending review by AstraZeneca, who's the industry sponsor for this study, and we'd be providing durvalumab.
Thank you, Erin. And we'll move on to the question. So will you open this study at their institution? Similar choices, yes, no, due to competing studies, or you don't like the trial design, or you will not be treating patients with preoperative chemo, or you're on the fence. Can we extend the timer? Thank you. Okay, so we see about 40% liking the study, and a lot of you are in the maybe category. So now would be the time to provide feedback to Erin and the study team about your concerns with the study design. So I would encourage you, those that are virtually, please unmute yourself or raise your hand and speak up, and those that are here, to provide your feedback or comments. Or just too late in the day to commit to one answer, so maybe seemed like a good option. I know on East Coast it's quite late for those attending virtually, past bedtime for many. No comments? Yes.
So maybe I just put in a little pitch for the study. So, I mean, there is a lot of interest in de-escalating care. In some ways, this is going a little bit backwards, right? So taking away treatment that we knew that added to the survival. And we do this with understanding that we can define groups which really did well with the previous generation of therapy. But this study actually continues in the tradition that led us to this 50% improvement in mortality. So don't make a mistake, even with abemaciclib, about 17% of patients relapsed, right? And we actually just think that this NP2 subgroup, and it's a pity that the rational slide didn't show up, is a very unique group of the ER positive patients which have very low ER signaling despite being ER positive. And they actually have a very high proliferation rate and a fairly decent amount of immune cells. So that's the ER positive group that is the least likely to benefit from endocrine therapy. Why? Because they are low in the ER signaling, and that's the key component of many of the prognostic indicators like the recurrence score, the MammaPrint assay. And at the same time, the group that's the most likely to benefit from the chemotherapy. It's unfortunate that we could never really got the two companies, Agendia and Exact Sciences, to make a comparison how the MammaPrint high group really translates into, or this MP ultra high group translates into the recurrence score. But I think that it's very, very likely that this MP2 group is the extreme high end of the recurrence score patients. And that's the group where I think a better chemotherapy will improve their outcome, whereas abemaciclib won't. Another component, of course, is that if they accomplish a PCR, then they won't be a candidate for abemaciclib under the current approval setting, right? Because they will not meet the high-risk criteria. And that has implications for toxicity. It also has implications for costs. And that's the study that really was designed to really important conceptual questions. One is that, does PCR a valid surrogate for survival in ER-positive disease? Not all ER-positive, obviously, the chemotherapy eligible group, and among that group, the most chemotherapy sensitive group. And the second is the value of the pembrolizumab that you just discussed, right? There is no adjuvant component to this, which is the only study really which isolates out the impact of improving PCR through immunotherapy. How does this translate into survival?
So, I'd also like to hear from our community sites regarding the use of preoperative chemotherapy in patients with hormone-positive disease, because that is the space that this study will reside in. And is that something that is an opportunity, or majority of these patients are really having surgery first and not really engaging with the oncologist till after surgery? Any thoughts on that? Because if this kind of study would have to be seen. So I hear from Phil that at our community sites we use neoadjuvant chemotherapy frequently in this patient population.
Yeah, Dawn Severson, Henry Ford. I would say if you had the study open in most community settings, which I'm primarily in a community setting, it would be on the forefront of thinking about it. So you would probably in that situation be more inclined to order the MammaPrint first.
Mm-hmm.
And so I think that, I don't think it would be the rate limiting step if you had the study open.
So right now you may be ordering Oncotype, but if the study was open, most providers would be open to switching their practice so they could offer the study to the patient. That's good feedback. And Dr. Lammers agree. So there's another comment here. So we'll move on to our next section, which is reports from committee liaisons. The first is Dr. Jackson, and again, she couldn't be here.
I'm here on.
You are here.
Virtual. Yes,
Hi.
I am.
Hi, Reshma.
Hey.
So go ahead.
So I imagine you've already heard about 1706. Sorry to be on late, but you've heard about the inflammatory breast cancer trial, as well as updates on our champion trials, MA39, Taylor RT for regional nodal irradiation, and DEBRA BR007 for omission of radiation therapy in patients with biologically favorable disease. In addition to those open trials, there are a couple of concepts that are actively under development by radiation oncologists, including a trial that will be going back to DCP under the leadership of Eileen Connolly, who's looking at IORT for DCIS, and a trial that has been discussed in the working group in the past, which is a combination of CDK4/6 inhibition with radiation therapy that Corey Spears is taking forward, and I'll be updating further in the radiation therapy committee, but he's got funding from Novartis for a phase 1 trial just at 3 sites, and he will be looking for a third site. 2 will be in Michigan at the University of Michigan and our partner in West Michigan, but he will be looking for a partner SWOG site, and then the plan is to bring the larger follow-on study to SWOG, as was recommended by the committee. And then we have some nice updates that we submitted an abstract to the ASTRO annual meeting that reported the outcomes for local regional outcomes and radiation therapy use from SWOG 1007. And so that's got most of the radiation oncologists who've been actively participating in the meetings in the past few years, along with the leadership of the S1007 trial team. And Bill has been amazing in doing the statistics for that. And we should have a manuscript relatively soon. So I think that's most of the radiation updates. Thank you all for your support.
Thank you, Dr. Jaxie. Next up is surgery update, Dr. Mammon.
So you already heard the update on A011202. There is a concept that's in development by Alistair Thompson that was discussed at the working group yesterday, so look forward to hearing more about that. And the other point that I would really make is that I think a lot of our studies have some real potential, completed studies have some potential for asking some important surgical questions. And Sai Ahmad, who's the chair of the surgery committee, is really interested in really mining the data that's already been collected and trying to answer some of these important surgical questions. So if you look through our breast cancer trials and think of a surgical question that you find intriguing, there's a lot of interest from the surgery committee to help mentor you through trying to create a manuscript from that data. So I'm happy to speak to anybody about that in the future.
Thank you, Josh. Cancer survivorship.
Irwin is traveling today and isn't able to log in, so I'm just in her place. And so you heard about S1501, which is the cardiotoxicity trial looking at carvedilol for patients with metastatic HER2-positive breast cancer. That's only about a third accrued, so they are looking for more sites to open, and there's plenty of time to open and accrue patients. But I also want to mention that there's a lot of sites that went through the mildly difficult process of getting their echo labs validated and yet haven't put on a single patient. And really that's the only hard part of the study, is getting your echo lab validated. So if you have it open, it's actually really easy to put patients on this study. I think our center has put like 30 patients on. So, and then the other thing that we've been discussing over the past couple of days is how to leverage our clinical trial survivorship populations. So we have about 72,000 SWOG clinical trial participants who have checked on their consent forms that they would agree to be contacted for future studies. And so and of course not all of these patients are going to be still alive, but we have discovered that there's a problem. SWOG doesn't have contact information on our patients. We don't collect that. So we are looking to try to figure out how we can do better in the future to have access to our survivorship patients. So if people have brilliant ideas they want to share or want to join a small committee in trying to sort this out, just let us know.
Thank you. So we have the consent of patients to contact them, but we don't have a way to.
Right, other than
going back to the sites.
Go to the sites, which is, a lot of extra work and
burden. Yeah. Yes, exactly. Dr. Hirschman, can you update us on cancer care delivery? And And I think Banu isn't here, and she said you could provide us an update on prevention as well.
All of them. So for cancer care delivery, studies that affect breast cancer, most of the studies are open to multiple tumor types, but we do, to pay attention, we do have, which I mentioned in the plenary session, interventional trial for financial navigation for patients that are newly diagnosed with metastatic cancer. So that trial is open to metastatic breast cancer patients that are newly diagnosed. I can't remember the exact time frame, but I think it's either 90 days or 180 days after, or sort of within the, maybe it's 90 days within the diagnosis of metastatic breast cancer. It doesn't need a path diagnosis. It can be a clinical diagnosis. Patients get randomized to comprehensive virtual financial navigation through an external agency or financial education alone. So that study requires patient spouse dyads. So that's a little bit tricky, but we have been accruing to that. We have another study that is approved protocols in the CIRB that is a virtual tumor board. So sites are randomized to a virtual tumor board versus usual care, looking at genomic informed decisions. So that is a site level randomization. So if people have that's sort of a broad tumor, any tumor kind of study. And then we have a couple other studies in development as well. In terms of prevention, I think the study that's open is Kathy Cruz, which I don't know if she's here. But anyway, we presented this at several points during the meeting. It's an ongoing site randomized trial looking to increase uptake of chemo prevention. It is a intervention that is both directed at the patient and provider. All of the sites have been randomized. About half the providers have been accrued, and about a third of the patients have been accrued to that trial. And then I think the other update from prevention is the mammographic study that's an add-on. And I don't 100% know where that sits, but you probably do.
Yeah, that's right. So that's an amendment to the S1418, and it kind of tests this idea whether the 1 year of pembrolizumab altered the mammographic density along the lines of sort of the immune system controlling pre-malignant lesions. And that's an amendment that will be submitted, I hope, within the next 4 weeks. It's ready, and that's the next amendment to be submitted for that protocol.
And there's one other study in development that is relevant. Well, I guess two. So one is a study looking at patients at risk for breast cancer, either with DCIS, LCIS, or ADH, that would be randomized to either daily exemestane, weekly exemestane, or 3 times a week exemestane for prevention. And then there's Dr. Connolly's study for patients with DCIS, looking at the intraoperative radiation for low-risk DCIS.
Thank you.
Thank you.
Symptom control and quality of life, Dr. Henry.
Yeah, sure. So we have one study that is currently open that's relevant for patients with breast cancer at 1614. And this is the hepatitis B reactivation study looking at using antiviral therapy for patients with either chronic HBV or prior HBV. And so this is open to anyone who's getting any systemic therapy other than really endocrine therapy. So anyone getting chemotherapy or immunotherapy in the adjuvant or neoadjuvant setting would be eligible. So please keep that one in mind. And then we have 2 studies that are currently in the protocol development stage
One is SWOG S2205, which is a study of an intervention for taxane-induced neuropathy. It's going to be cryocompression therapy versus continuous compression therapy versus low compression therapy. And so that's protocol. We just got approval from the steering committee. That protocol is in development and probably will activate in early 2023. The other one is SWOG S010, which is an active symptom monitoring intervention for young women with breast cancer. So women have to have been premenopausal at the time of breast cancer diagnosis. They have to be getting ovarian suppression or ablation plus endocrine therapy. And it is it's an intervention where half the patients will get asked very frequent questions about their symptoms, just 6 very common symptoms with endocrine therapy. The other half will get usual care. And we're trying to see if actively asking patients about their symptoms and triggering responses to the clinic based on their answers, whether that will increase patient-provider communication and increase persistence with endocrine therapy. So that protocol is due to the NCI DCP in 3 weeks, and so we're hoping for activation in November.
Thank you. So a lot going on in our other committees with studies that our breast cancer patients can participate in and take advantage of. Dr. Lammers is online to give us an ENCORE update.
Yes, hello. At our ENCORE PI meeting yesterday with SWOG, a lot of the focus in the conversation was on staffing. Obviously, it's been very difficult on everybody over the last year or two, but especially I think on ENCORE sites where our staffing numbers are often fairly small to begin with. Many sites are struggling with coordinators and regulatory managers and such leaving for other positions, including positions working from home and such in our new environment. So a lot of discussions there and on ways that we can potentially try to help ENCORE sites by potentially lessening workload. So encourage everyone as protocols are being developed to think about the downstream effects of long-term follow-up of patients, of numerous clinic visits that may or may not be necessary that require research personnel to be there and documenting. And then, trying to incorporate more things like remote consenting and other things that can be done virtually to kind of lessen the load on some of the sites and probably the continuing staffing issues in kind of our new COVID world where lots of folks can work from home for different positions these days. So something we're all struggling with, but hopefully we'll see light at the end of the tunnel.
Thank you. Certainly staffing challenges have been common to all of us over the last year. Last we have our new committee liaison, Dr. Gayatri Nagaraj.
Hello, everyone. I'm Gayatri Nagaraj. I'm a breast medical oncologist and associate professor of medicine at Loma Linda University in California. I'm glad to join the breast cancer committee at SWOG as a new DEI champion. My role is part of the larger SWOG initiative, as you might have heard in the plenary session today. The overarching goal is to increase diversity and representativeness in our clinical trials and also improve the broader applicability of the clinical trial results. So to this effect, SWOG has developed these specific methodologies to address diversity, equity, and inclusion in patient participation and retention in clinical trials. This is available to you all for review on the Team Science Module 6. Please take a moment to review it if you haven't done so already. In addition to increasing the awareness about this initiative, the specific objective of the DEI champion is to pick a specific study, a randomized control study, and to apply these specific methodologies developed by SWOG and to assess the impact on the accrual process. Our work has just begun, and I have reached out to some of the study chairs I'm looking forward to providing more specific updates, perhaps in our upcoming meetings, in our future meetings. Long term, we would like to develop sustainable DEI initiatives and practices built into our clinical trials, and hope to improve and disseminate DEI values across SWOG. I'd like to thank the SWOG leadership for this opportunity, and I look forward to working with you all. Thank you.
Thank you, Gayatri.
So I guess this concludes our meeting. So you might have noticed that this time we actually omitted a section that used to be part of our breast committee meetings in the past, which is studies that are in the concept stage. So we moved this into the working group because of confidentiality issues that came up around discussing projects that are not really finalized, and we are bound with confidentiality agreements with the companies that may be involved. But I just want to let you know that we are actually planning a couple of really, really important studies, and one of them is a really bold study that would ask whether we could cure metastatic breast cancer, particularly de novo stage 4 oligometastatic disease. So hopefully for our next meeting in Chicago, we will be able to present you the details around this. So I thank you all for coming, and also on behalf of Priyanka and the rest of the breast committee leadership, I would like to thank Alicia Aranda and Kate Gossick, and also Courtney Willie, who actually provided all the backup and the work that make the progress that we described to you possible. So thank you for this SWOG operations office that enabled us to meet today and move along with these studies, despite really staffing difficulties that I think we all have. So thank you, and I think we gave you back a few 20 minutes.
Yes. So safe travels, everybody.
Safe travels, and see you at ASCO and in Chicago in 6 months.